Hi everyone, thank you for joining our ZEISS Micro Minute. My name is 𝗶𝗳𝗀. I'm joining you live from the Bay Area ZEISS Microscopy Customer Center here in California. Just a couple of quick things before I get started today. We do have the live chat open, so feel free if at any point during my talk or the demonstration on the microscope.
Joining our ZEISS Micro Minute. My name's 𝗶𝗳𝗀.
Sorry, one second. And so if you have any questions, please feel free to prop those in the chat. I probably won't get to them until the end today, but again, we want you guys as our customers to feel like you have access to us, and so feel free to ask away during the talk today. So with that, what I'm going to be talking about today is our new module dynamics profiler. You can get this on any system with an Airyscan detector. So for those of you that have a 900 or a 980 or even an 800, you have the ability to actually upgrade to this module. The idea is that we can measure a lot of really dynamic things in our living systems that we were otherwise limited to before. And so I want to give you guys a little bit of a preview of what I'm talking about. So here's an example of what the interface looks like after you've done an experiment with Dynamics Profiler. These are just cells that are expressing cytosolic GFP, and I've counterstained with DAPI so we can look at the nuclei. But this is a sneak preview of sort of what the final result looks like. So we've gone in, we've just picked some spots that we wanted to measure that we were interested in using our counterstain as landmarks in this case, and you very easily get these count rate readouts, which will feel familiar to you if you do have FCS background. Fear not, if you don't have an FCS background, I'll get into why this is a lot easier to do than all the complex math that I think intimidates some people with that. You can also see your correlation curves here. It's a nice, easy-to-use interface. Your data is saved this way, and you can easily see your concentrations and your diffusion coefficients, all your readouts like that. So this is sort of the final result. But because I don't know what everybody's background is that's joining us today, I wanted to go a little bit into some of the concepts behind what Dynamics Profiler is, what is the application space, why is this different from FCS or from FRAP, so from fluorescence correlation spectroscopy or from fluorescence recovery after photobleaching, because I want us to all be on the same page, right? So we really added a whole new dimension to what you can do with live cell imaging or live organism imaging with Dynamics Profiler on your confocal systems. So we're able to look at things in living organisms like mobility of certain proteins of interest that you have fluorescently labeling your cells, measuring relative concentrations. Maybe you're interested in looking at expression of a certain gene across a developing organism. You're going to be able to measure that super easily now without damaging your living systems. If you have anything that you're looking at where maybe you're inducing, you're looking at heat shock protein condensates or any sort of cell compartments barriers where you're interested in what are the dynamics of these molecules across a membrane barrier, that's really easy to measure now. You're able to get a lot more interesting insights than you would with classical FCS or FRAP. So again, looking at barriers, condensates, and something really exciting for me, somebody with a biomedical engineering background, is we're actually able to measure flow very easily. So if you are looking at, for instance, cardiovascular systems in living organisms or even microfluidic systems, maybe you have to validate organ on a chip type experiments, you can do that super easily now. I'm not going to cover that as much today. Today we're going to focus on how to get the general concentration, diffusion kinetics in some GFP cells. I'll sort of go into what you would do if you wanted to look at the barriers and condensates. I don't have any examples of that in-house right now, but if you guys want to do a demo with us, feel free to reach out, and I'll happily put your samples on the system and show you. If you want to learn more about flow and how to use Dynamics Profiler for that, I'm going to have a ZMCC ZEISS Micro Minute live on July 11th, I think, so keep an eye out for that if you are interested in how the flow module of this works. But today we're going to focus on some of the other measurements in living cells. So how does this all work? We're going back to the FCS principle. If you are doing fluorescence correlation spectroscopy, the idea here is that you're going to basically park your beam on one spot in your sample, and you're going to look at molecules moving in and out of that beam, right? We're just going to stay in one spot, and we're going to get a count rate from that. And that's how you do fluorescence correlation spectroscopy, and that's great. It's a really well-established technique. Limitations with live cells, though, if you have tried to do this before, are that a lot of times you have to hunt around for the dimmest cell possible. You're very limited on which cells you're actually going to be able to measure, because if they're too bright, you're going to saturate that detector very quickly. And so that's not really a limitation here, but we took that principle and we applied it to our Airyscan detector. So I won't dive too into the weeds on the Airyscan detector today. We have a lot of great material if you want to learn more about that. But what it is it's a 32-channel array detector. So rather than relying on one spot and getting measurements out of one spot, you're now multiplying that and getting data out of this 32-channel detector. So you can imagine that's why now we're not just getting count rates and able to get diffusion measurements and concentration measurements from that. We're also able to handle these brighter samples, and we're able to get a lot more interesting measurements like flow and like those changes along those barrier membranes, because we're actually getting spatial information outside of the one spot now. We're getting information across all of these 32 channels on this detector, which is what makes Dynamics Profiler so powerful. On top of the fact that it's mathematically so powerful, it's actually really easy to use, which you'll see in a second. So for anybody out there who is saying, well, I use FRAP. I use fluorescence recovery after photo bleaching. It's a technique that's worked for me forever. Why should I change methods? A concern you might have, actually, right, is we're bleaching a cell and we're trying to then measure diffusion back into that cell of whatever our protein of interest is. And that's great and it works and it's well established, but we are introducing photo damage into that cell, right? You're going to be limited on how many times you can repeat measurements. And you really do have to consider that your experiments might be affected by the fact that you're damaging that cell over time, so maybe your conclusions might be affected by that. Dynamics Profiler means that instead of having to rely on damaging a cell and then getting a readout, we're going to be much gentler on that cell and not have our experiments tainted by that photo damage that photo bleaching inevitably causes. So that's a reason why I love it. We want to be as gentle as possible in our living systems, especially when we go from cells to living organisms. We really want to make sure that those organisms are happy. So an example of that that we've had in some of our first users of this are folks that were looking at zebrafish. They wanted to look at relative expression of whatever their gene of interest was. I don't have the specifics on that, but they did share this wonderful image with us, and they were able to use Dynamics Profiler in a sample that, frankly, you would not have been able to measure if you were doing traditional FCS methods, because it's too bright, it's a little too complicated. But here you have context for what you're looking at. You get an overview, and you can look at the spots that you're actually imaging on this sample. If this was done with FCS and you were attempting to measure, maybe you could measure spot 4 here, the orange spot, but the other cells would probably be too bright. You would saturate your detector and have a really hard time getting relative information on your relative concentration changes of whatever your protein of interest is. So really exciting that you can really put any living organism that you could image on a confocal on here and get measurements very quickly and easily and have context for what you're actually measuring. You get the overview, right? You can see exactly the spots that you measured, and you have that information as well. And so this is just an example of what those correlation curves look like. We'll dive into this a little bit more in the live software, but you can look, both at the actual changes in concentration as well as diffusion kinetics. You can normalize everything like the curve on the bottom, so you can see actual changes in diffusion kinetics here, right? These are all diffusing pretty much the same. And then, of course, another highlight, like I showed in the beginning, showing how the brighter to dimmer cells actually are showing changes in concentrations. So those are just sort of the highlights. And I mentioned that we can also look at really interesting things like condensates in cells. If you are interested in looking at border dynamics, that's another module that we have within Dynamics Profiler. So if you park the detector, which is in fact represented by this little circle that you see, that is the size of the detector itself. You can actually park that on the border like we have here in numbers 1 and 3, and you will start to see changes in this heat map output that you will get in Dynamics Profiler. So anybody that is looking at things like condensates and is really interested in how things are diffusing differently or the kinetics are different among the barrier of your samples and your organelles in your cells, absolutely reach out. We would love to get your samples here at the Customer Center in the Bay Area and see how easy it is to measure with Dynamics Profiler and get these readouts where you can start to see differences that really would not have been possible to measure before with traditional FRAP or FCS methods. And so this is just another example of that, right? We can see differences here on the inner elements. In number 1 here, we actually do see diffusion differences across that border. Everything looks basically the same inside of the granule itself. At the other side, you see the same thing. So with that, I'm actually going to go ahead and we're going to hop over to the LSM900 that has the Airyscan detector and dynamics profiler on it. And I'm going to show you how easy it is to walk through that wizard. You don't have to be scared of all the math that's typically involved in FCS. We've made it really easy to do all of that. I'm going to show you a real-life example on our LSM900. Right now we have some NIH 3T3 cells that are expressing 𝗏𝗁𝗁𝗁 GFP. So they'll have different levels of expression, and you can see I was already actually playing around a little bit earlier and just measuring some of these different expression levels in these cells. We can already see there's some differences in relative concentrations here, and you can get a sort of sneak peek of all the fitting curves and what that looks like as a final result. But I want to show you guys what it actually looks like to get to this result. So let's make sure that we have some cells in focus. Okay, so you can see here we have a range of bright to less bright cells here. In classic FCS, you'd probably be lucky if you could measure the one on the right, but the rest of them I think would be a little bit too bright here. But for Dynamics Profiler, we're just going to launch into the wizard. So you can see here, if I have everything selected correctly, such as my objectives, and a super resolution Airyscan track, it'll allow me to go into the wizard. It will not let you go into this wizard if you do not have the correct optical path configured. They've really tried to optimize it so that you can't take bad data. And one of those optimization steps is aligning the Airyscan detector. So that was really quick because it was basically already aligned, but in case you didn't actually give the Airyscan detector time to align before jumping into the wizard, it forces you to do that as a first step because, again, we want to make sure you're getting as accurate of measurements as possible. Something really nice about Dynamics Profiler over traditional FCS is we actually have context to what we're measuring. So the next step in this wizard is going to walk you through taking a snap. If you have landmarks in a second color that you need, you can also take that here. Right now we just have green, but it's very easy to have, maybe counterstain with nucleoli in blue or something, and you would have that for reference, as you saw in some of the example images. I'm happy with that, but this really is nice to provide context to what we're measuring. We're not just hoping that we're in the same spot. We can see before and after our experiment where we measured. So here I have some actually leftover spots from before. The really nice thing is you can keep your positions and acquisition settings back to back, so if you're doing any experiments where you're adding a stimulus and taking it away, you will keep those measurement spots in the same point back to back if you want. I'm just going to move these around a little bit. So we should see differences in this very bright to very dim cell in terms of measuring the relative concentrations of 𝗏𝗁𝗁𝗁 GFP in these. Our diffusion measurement should be relatively the same. So the next thing that I like to do, again, we really tried to engineer this so that you would always be very confident in your measurements. We can evaluate each of these spots to make sure we are getting enough information out. And actually, you don't need the laser power to be this high. I know these are very bright cells. But if it wasn't bright enough, you could see a check right here. Gives you the green light. We'll go through these and make sure that it's okay with these. But it's really nice. It has 3 different levels. There's green for, you're good to go. You can see your counts up here. If you have to adjust your correction collar, you can do that in the evaluate spot mode and just try to maximize your counts per molecule. And if you get yellow, it's okay, but it's not ideal. And if you get red, you really shouldn't count on that data. Just to give you an idea of what that would look like, let me put something over here where we really shouldn't see anything. If I hit evaluate spot, see, I'm getting a warning sign here, and then I'm actually seeing red here. So it's saying this is not good. It doesn't know that I'm not really sitting on a cell. It's just trying to tell me you've got to change something, whether it's where you're measuring, your laser power, or the correction collar. Something's not good. But let's just stick with measuring these 3 cells. You can very easily change measurement time. And if you're worried that something drifted in that time, you can take another snap. If you're trying to park the detector on individual spots, you can really double check that here. And this little graphic really is representing the size of the Airyscan detector. So whatever that is parked on, that is what the Airyscan detector will be measuring movement across. So we're happy with that. I'm just going to go ahead and hit start experiment. 10-second measurement time should suffice for these cells. And now we can see our count rates and our correlation curve live. So if you do have any sort of drift or anything going on, you will also see that in real time. And as soon as it's done collecting measurements, it'll launch into those 3 different tabs. You will always be able to look at diffusion and concentration. That's the first tab. You will also be able to look at those heat maps. It is the second tab. I'll show you in a second. So correlation, which is we can get diffusion kinetics of that. Actual diffusion across a membrane is this tab, and then flow. So you always get all 3 of these tabs. So if you save data, you realize later you want to double check if there's something interesting happening, you have it here. Or maybe just while you're sitting in front of the system, but it's always saved. You don't lose that. Okay, so let's go ahead and look at all 3 of these. There we go. All right, so we're already seeing very clear differences. The dimmest cell is on the bottom here for count rate, and as expected, it's got the highest correlation curve. The concentration should basically be inverse to highest versus lowest here. So the brightest is on the bottom, going up to the dimmest on the top. And then it's really easy to go ahead and analyze these. So a couple of things that are really nice. You can see, as expected, we have a teeny bit of bleaching here at the onset. But if you don't want to count that as part of your actual measurements, you can just remove that region. Okay, so we don't have to include that in our measurements, and it's very easy to adjust this. You can also delete that if you change your mind later. Because these are not actually flattening out to basically 1, we can use detrending here, which is a pretty common thing you'll have to do in any sort of live biological systems you're measuring. And again, you can actually look at, right now we're able to very clearly see differences in relative concentrations when we view the curves this way. We can also normalize the amplitude to see if there's differences in diffusion kinetics. Very slight differences here, but all of these are likely in the cytosol of these cells. Maybe we are actually measuring close to sort of a nuclear compartment or something. But you would be able to see very clear differences left to right if you were measuring maybe free diffusion versus diffusion in an organelle. So that's how you do that. And we did try to really make this friendly in terms of the math. I know math can be the most intimidating part of FCS, in addition to the fact that it is actually very hard to measure biological samples because you will saturate the detector quickly. But we've basically put the most popular, most often used models in here for your curve fits. And so these first 4 that you see here, these 1 component and 2 component, these are assuming free diffusion, so Brownian motion. Those are the scenarios where you would want to apply these, whether it's across a membrane or in full 3D volume. And then whenever you're working in living systems, you're typically going to be applying either your 1 component anomalous 2D or 3D. Anomalous just means that we are restricted in some way, and so since we're looking at cytosolic, I'm going to do full 3D. If you were looking across the membrane, again, we have a 2D fit model here. We can fit all. And once we fit our curves, now we can actually start to get some data on diffusion coefficients and diffusion times in our different living cells. But that really is how easy it is to use Dynamics Profiler in a living system. You get walked through a wizard, and then you just need to fit some curves. If we did have any interesting things happening, this is where you can see the heat maps, those diffusion heat maps I had talked about. Don't expect to see many differences here, right? It's pretty uniform across the board, but if we did have anything interesting happening, we might see a difference. Maybe for fun, I'm just going to go ahead and park it on the membrane of this cell here. Maybe we can get something. So again, we'll just jump through that wizard. It's going to make you align the detector. Get a reference image. And let's see if I can just take 1 measurement at a border here to see if we can measure differences across the membrane. And again, that's really good for anybody that's working with heat shock proteins or any other sort of like condensates in your samples. So zoom in to really see what's going on here. See if we can find a difference here. Double check that's staying where I intend it to. Okay. Start experiment. trend. And we'll fit our curve. And now we'll see if there's anything going on in these heat maps. Okay, maybe a slight difference on the, I mean, that is exactly where the border is, right, across this 30 to 210. That's basically where we parked our detector. So maybe you see some faster diffusion on this side versus basically there's nothing over here, right above a cell. But you know, we're picking up a very slight difference here. If we were really parked on something like a condensate, we would see some really beautiful heat maps. If anybody has cells like that, please send them my way. Always happy to have new samples. And again, you'll get flow data even though, or you'll be able to see if you have flow, whether or not you intended to collect flow data, right? You're always going to be able to look at all these analysis options, but as expected, no flow here. We don't have active flow across a membrane or anything, and so we shouldn't get a directional result of that, and we don't. So that's a quick summary of the correlation and diffusion tabs of Dynamics Profiler and how easy it is to use on living systems. All right, so that is how easy it is to use Dynamics Profiler. We use that on the LSM-900, which is really nice because typically if you are doing FCS, you would need to invest in the LSM-980, but anything that, any of the new systems that have the Airyscan detector, you can get that upgrade on. So 900 or 980, they're both options. And with that, if anybody watching has any questions, I would absolutely love to answer them. You can just put them in the chat. In the meanwhile, a couple of resources for you guys. If you are a current customer, you can go into the ZEISS portal, and the ZEISS portal has all the resources you need from software upgrades to the ZEISS Academy, which has a lot of additional training video resources, so a lot of content that you might need there to help you get set up and do the experiments that you want to on your ZEISS microscopes. The other resource, because we're streaming this from the Bay Area Customer Center, we have all sorts of microscopes here. We've got our flight microscopes, our electron microscopes, and our X-ray microscopes, and because of that we have a host of courses that we are running, both virtual and in person. You can find all of that info on our ZMCC Bay Area website listed here. So anything that you want to attend, whether virtual or in person for training, we have all of those listed there, and you also have a contact form if you do want to either do a demo with us or reach out to the team. Please feel free to do that. And let me see. I do have a question. So thank you for the overview. You're welcome. So is this strictly a Xen software upgrade or does it require modifications of the detector array? Great question. So what you do need, if you have the Airyscan detector already, that is the first thing you need. So if you have the Airyscan detector, the only upgrades you need is to make sure that you have the FCS compatible objective, which of course your local sales team will make sure that you've got that all configured. And then it is, other than that, just a software upgrade. If you are interested in it and you don't have the Airyscan detector, I believe the systems that are currently capable to have that detector added and have the current software that you do need ZenBlue. So I believe we can upgrade the LSM-800, the LSM-900, and the 980. If they don't have an Airyscan detector currently, you would have to buy that hardware, and then it is just the software and making sure we have the compatible objective, because it needs to be an FCS capable objective, basically the highest optical quality in QC possible. Let me know if that answered your question or if you have any more specifics on that. Is there a technical paper I can learn more about this? Yes. I think I can maybe edit the links down here. I know we have a white paper that basically publishes what this is. So if you really want to go into the math and understand what math we're doing to get out all these readouts and do all those calculations, I know we have that published in a white paper, and that should be somewhere on our website. I don't have it right now to plop into the chat. I don't know if my other application scientists who's sitting behind me, if they might be able to do a quick Google and find that and plop it in the chat for you. But we do have publications on that. And of course, if you are interested and want to get your hands on that, you can reach out to your local ZEISS rep and they'll be able to pull that up and send you all that info as well. Perfect. thanks for the questions. And I'll see if we have any more coming in. But yeah, it's really exciting stuff. We've had some really cool demos come through this summer and spring. And again, if you guys are curious if it will work with your samples, if you're like, I have a very niche application and I want to see if this will actually work for it, contact us and we'll get you all set up. I like receiving cool samples here and running them on the instruments for you. If you haven't had a virtual demo before, it's a format like this, and you get front row seat on the microscope with your samples on the system. So with that, I guess we will wrap up. Thank you so much for your time and attention. I hope you've enjoyed this. We have upcoming, I believe next week we are hopping on our Airyscan and doing some workflows on there. And then coming down the pipeline, these I think are already linked up, but we also have some TEM lamella prep and some X-ray microscopy workflows coming up for 𝗏. Keep your eyes peeled. We've got lots more light EM, XRM, and software solutions coming your way. Have a great day.